Assalamualaikum warahmatullahi wabarakatuh. My name is Khoirun ulfi. I am from Walisongo State Islamic University, Semarang. In this occasion, I will do an experiment with the title is the effect of visible light spectrum and soaking time on mung bean seed or Vigna radiata L germination process. Mung bean or Vigna radiata L are a type of legume that is widely cultivated. The development of mung bean starts from the germination process. Spores are small plants that have just emerged from seeds, and their leaves still depend on the food supply contained in the seeds. The spores will grow and develop into seedlings, which in the next stage will grow an adult plant. The seed germination process is influenced by internal and external factors. Sunlight and water availability are examples of external factors that will be tested in this experiment. Light is the main source of energy for photosynthesis and growth. Visible light is the portion of the electromagnetic spectrum that can be detected of various colors by the human eye, ranging in wavelength from about 380 nanometer to about 740 nanometer. Differences in light characteristics such as spectrum composition or wavelength, intensity, duration, and direction of light influence plant growth and development. The composition of the color spectrum of light can be manipulated by filtering used colored plastic as a filter. If light is visible through a transparent plastic sheet, the radiation that is released will have the same color as the plastic. So, the plants under the plastic will receive radiation according to the color of the plastic. Meanwhile, the length of soaking will affect the germination process. Through the imbibition process, water will facilitate the process of destroying the seed coat and make it easier for the seed to absorb nutrients in the growing medium. From the theoretical basis, the purpose of this experiment is 1. To determine the effect of various visible light spectrum through colored boxes on the germination of mung bean seeds. 2. To determine the effect of soaking time for mung bean seeds on the germination of mung bean seeds. The tools and materials are mung bean seeds, water, cotton, plastic cups, plastic box, red, yellow, green, blue, and violet plastics, roller, hand pond, whiteboard marker, syringe, scissors, and tape. Ways of working. 1. Prepare the tools and materials that will be used. 2. Soak the mung bean seeds in a plastic cup with varying soaking time of 2 hours, 4 hours, and 6 hours. 3. Put cotton in a plastic cup as a growing medium. 4. Mung bean seeds that have been soaked for 2 hours are put into 5 plastic cups filled with cotton, 5 seeds per cup. This is also done on mung bean seeds that have been soaked for 4 hours and 6 hours. 5. Each plastic box is coated with one colored plastic, resulting in red, yellow, green, blue, and violet colored box. 6. Each box is filled with 1 cup of mung bean seeds which are soaked for 2 hours, 4 hours, and 6 hours. So, in one box, 3 plastic cups. 7. Mung bean seeds are watered once a day with 10 ml of water for 4 days of treatment. 8. Mung bean seeds are counted every day for their germination lengths using a ruler. 9. The data obtained were graphed and analyzed which visible light spectrum and soaking time were most effective for germinating mung bean seed. The main observation parameter is plain height, measured with a ruler with an accuracy of 1 millimeter. The following are the results of measuring mung bean seed spores based on the length of soaking and the exposure to different visible light spectrum. By soaking mung bean seeds for 2 hours, the results show that the red light spectrum with a wavelength of 625 to 740 nanometer was the best for the germination process. This was proven by the final result that the length of the spores in the red box on day 4 was the longest, namely 1.1 cm. Meanwhile, the mung bean seeds in the violet box are the lowest, namely 0 cm, or do not germinate. This is because mung bean plants contain phytochromes, namely proteins in chromatophore that are similar to phytocyanin. Phytochromes in mung bean have a reversible structure that can absorb red light energy according to the light needed for the growth of mung beans. Holiday and Carnot, 2016. By soaking mung bean seed for 4 hours, the results show that the yellow light spectrum with a wavelength of 565 to 590 nanometer was the best for the germination process. This was proven by the final result that the length of the sprouts in the yellow box on day 4 was the longest, namely 1.8 cm. Meanwhile, the mung bean seeds in the violet box are the lowest, namely 1.3 cm. By soaking mung bean seeds for 6 hours, the results show that the violet-like spectrum with a wavelength of 380 to 425 nanometer was the best for the germination process. This was proven by the final result that the length of the sprouts in the violet box on day 4 was the longest, namely 5 cm. Meanwhile, the mung bean seeds in the yellow box are the lowest, namely 1.8 cm. If the germination speed is compared between the soaking time factor and the given visible light spectrum factor, the result is that the fastest germination occurs in mung bean seeds with a 6-hour soaking treatment and placed in the violet visible light spectrum, namely with a wavelength of 380 to 425 nanometer. The germination speed reached 2.1 cm per day. Meanwhile, the lowest germination speed occurs in sprouts that were soaked for 2 hours and placed in the violet visible light spectrum, namely with a germination speed of 0 cm per day or did not germinate. The conclusion in this experiment is 1. The visible light spectrum through the colored bird affects the germination of mung bean seeds. The red visible light spectrum with a wavelength of 625 to 740 nanometer is best for germinating seeds soaked for 2 hours. The yellow visible light spectrum with a wavelength of 565 to 590 nanometer is best for germinating seeds soaked for 4 hours, and the visible light spectrum violet with a wavelength of 380 to 425 nanometer is best for germinating seeds that are soaked for 6 hours. 2. The length of time soaking mung bean seeds affects the germination of mung bean seeds. Soaking seeds for 6 hours is the best time to speed up the germination process of mung bean seeds. That's enough for me. I hope it's useful. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.